I'm Teresa Mangum. I'm the director of the Obermann Center for Advanced Studies here at the University of Iowa. And one of the things that we're doing this year is a series on what is research now. So as we've been talking to scholars, artists, and researchers on campus, we've realized that we have a reward system and we have in our minds often assumptions about what we mean by research. But many of us are working quite differently than we did when we started the profession, or than that assumption would support. And so we're holding a series of sessions to just get a better understanding of the nature of research in different areas. And also in the course of that discussion, to think about what do we need from our evaluation systems to take fuller account of things like the group today, which is a wildly collaborative interdisciplinary group where people are doing very different things to contribute to the research. I'll just quickly note our speakers and then invite them to introduce themselves more fully in the midst of the conversation. So Ted Abel is Professor, Chair, and DEO of Neuroscience and Pharmacology. He's also the Roy J. Carver Chair in Neuroscience and the director of the Iowa Neuroscience Institute, among the many, many other hats he wears here at the University of Iowa. Jocelyn Cho is an assistant professor of internal medicine with an emphasis on pulmonary, critical care, and occupational medicine, and director of the Iowa Inflammation Program. Nanda Kumar Narayanan, who is working on getting in through the webinar system, and he'll be with us soon, is an associate professor of neurology and the Juanita J. Bartlett Professor of Neurology Research and Vice Chair for Basic and Translational Research in the Department of Neurology, again, among many other titles that he holds. And finally, Stanley Perlman is a professor of microbiology and immunology and professor of pediatrics here at Iowa. And his lab is working on, among other things, coronaviruses and SARS. So thank you. You're all such busy people. I'm so grateful to you all for being here today. And I'm just going to jump right in and open with a question. How have you built your team, and what is the collaborative process to pull together researchers doing such different kinds of work around a problem? Ted, you want to start us off?
Sure, I'm happy to. So I think it's an interesting challenge in the sciences. You'll hear about that today, and I think we can see those challenges by two papers that were published actually last fall in the journal Nature. One paper was a paper published by Paul Nurse, who won the Nobel Prize for his discoveries of the molecules that regulate the cell cycle and cell division, with implications for our understanding and treatment of cancer. And Paul wrote a one-page paper in Nature on the need for ideas in science. On the other side, we have a paper that came out in November. Actually, we have 17 papers that came out in November in Nature, with some 275 different authors, supported by the Brain Initiative, and identifying every cell type in the motor cortex controlling motor behavior in the brain. So those two papers, one in September in Nature and one in last November in Nature, really summarize the kind of two sides of what we have in the sciences to think about. One in which we seek ideas to understand how the brain works, or how the lung functions, or how viruses infect cells, or how to treat viral infections, as Stanley and Jocelyn can talk about. And on the other side, we have amazing technology in the Brain Initiative to be able to study at the single cell level cells in the brain. And so I think we need both of those. And the first thing comes with a sort of big idea, I think, a big question. It's kind of not worth getting 275 people together to write 17 papers supported by tens of hundreds of millions of dollars if it's not an important question and a big idea. The next, really, in many cases, comes a technique, an approach that a field has. And then comes groups of people. And those are groups of people that really, there really is a sociology of science, not just a science of science. And we know that from some old time sociologists like Bruno Latour, who studied it some 30, 35 years ago. But it's still the case that it's people that do science, and it's those interactions. And so we have this mix of big ideas, new techniques, and then individual communications and connections. And all of that has gotten changed with the pandemic, particularly the connections. And also the technology. It's gotten harder to do experiments because of various regulation, various difficulties with the pandemic. Hopefully, we've been able to make some more ideas. I hope that's one thing I hope in all of this. So that's just a general perspective to start with, and we can hear from others about their views.
Anybody else want to jump in? And I'm also really curious about how your team formed and how you're sustaining your work together through Zoom Live.
So we actually were Zooming before the pandemic quite extensively, and my office and our meeting rooms are all set up to be able to handle Zoom, and those collaborations really extended across the world, from India to England to Germany to Australia and Japan. And so, those were built at meetings, at in-person meetings over decades, really. And so you wind up knowing people well, even though they live, 5,000 miles away. And that's something that's been much harder to sustain over Zoom because everything is scheduled. But I think it really has been about, connection, starting with people that you're connected to, and then kind of building those connections out.
Mm-hmm. And I'm guessing that all of you have probably been the lead on projects. But I'm wondering in this team or in other teams that you're working on, how you enter and participate when you're not the lead, how you weave into an existing collaboration. And if you wouldn't mind, maybe Jocelyn, you could start.
So I'm trying to think if there's an example of sort of, I guess, maybe coming into a different collaboration. I think I'll just sort of first start by echoing what Ted said, which is that the collaborations that we had that were existing relied on Zoom because of distance always. And I think it's much harder to sort of build those new collaborations now when you're not sort of meeting people in a forum that sort of facilitates free exchange of ideas over dinner, or sort of those, conversations that we all had after the sort of formal part of a meeting was over. So I think that has been a little bit more challenging in the era of COVID. In terms of sort of integrating into a team, I mean, I think, understanding what your role is in the team, understanding exactly what the team needs from you and what you can bring to the team probably is the first step. That's happened, I guess, in some of our COVID work, where we have sort of basic scientists or people doing advanced imaging along, but maybe they don't sort of see patients clinically and know what those patients are experiencing. And so I think, if your role or what you're sort of bringing to the group is slightly different than other members, then that integration happens pretty naturally.
Stanley or Kumar, is there anything you'd like to add?
Yeah, I would just, go on.
Please.
Yeah, I guess I would just say that for my point of view, things really accelerated during the pandemic because there were many people. I collaborated with various people, but really in small collaborations. But since the pandemic began, since I was one of the 5 or 10 coronavirologists in the country before the pandemic, still working on the virus, I did my lab and I became parts of lots and lots of collaborations. And a lot of these were pretty amazing because they were organized by either the World Health Organization or the NIH to investigate various issues involved with the pandemic. And it meant we have, I don't know how many meetings per week now, where we discuss new data and people bring data in that they just acquired 20 minutes ago, which would normally never, ever happen in a normal research setting. So we share data, and then the questions are so different because what Ted and Jocelyn are talking about, I think, are much more established processes that can go on at a reasonable rate. But with the pandemic, one, whether necessary or not, one feels that things should be done immediately. So there's all these groups that get together, and the main thing that people do is just sharing their data. There's not as much keeping data to themselves until it's quite ready to really be fully ready for a publication or for talking about at a meeting. And so that's been a really big change. And of course, the other part of this that I think is really great has been incredible sharing of resources, so that when in our case, a new variant arises, we can usually get the virus within days of when it first arises, because that's the way research has to move forward on the pandemic. And I think the Omicron, actually, this variant may show some of the good and bad points, because this variant was discovered in the end of November. And talking about papers in Nature, there were 3 papers with probably 75 to 100 authors showing how these viruses worked either in tissue culture or in animals, and show how they evaded the antibody response. So huge numbers of people getting together and co-authoring papers where each had only a small component. So the requirements for this are be willing to recognize that there is an important problem. The ideas are not so difficult. It's more getting the experiments done and making sure that they're valid, and then cross-validating them and not repeating what these other groups are doing. So a little different scenario, but coming out to the same point.
That's really fascinating. And I want to come back to in the same way, Ted, you drew that comparison between ideas and these broad collaborations. I would think right now that there is a difference between people working on immediate issues. And Ted, you and I talked earlier about how you get that research out fast versus longer term projects that are more that the research is being released at a different stage in the process. So I want to come back to how that's changing your work. But first, Kumar, you were about to jump in too. Do you want to add something first?
I was going to comment on it. I think I'm from Seattle, so we have this sense that the earth isn't solid. And I think you're talking to a bunch of people who, the ground has shifted underneath us, and it's hard for us to figure out exactly which way things are going to go. And, that ground is really driven by the pandemic. When I'm a Parkinson's neurologist, one of the most common conversations I have in my clinic is, when is there going to be a cure for Parkinson's? And before the pandemic, I would go into research, how it's done, how long it takes to do a trial. But now all of my patients are intimately familiar with trials, clinical trials, the fact that these scientists can be wrong. They know what preprints are. They know about preliminary data. They know that, science is not a straight process. And they know the simple questions like, should I wear a mask? When should I wear a mask? All these things are open to debate. And I feel like in the last 2 years, our scientific machinations are now open to the public in a way that we weren't sure of. And they're moving at a much faster way. If I had to talk to Dr. Abel or Dr. Perlman 3 years ago about posting things on preprints, they'd be like, well, what are preprints exactly? And now I'm sure they have no issues, like, let's get it out there. Let's get engagement and dialogue. And I'm hopeful actually that the things we've learned during the coronavirus as a society, that science is important, that research is important, that you got to invest in basic mechanisms, and that you got to study these in humans, those things are going to transfer, I'm hopeful, to my disease, to Alzheimer's disease, which is, we don't even know where to start with that disease. And I'm hopeful that these insights will transfer and improve the research process. Now, obviously, that's a hope. We got to make it happen. But, and it's going to need teams like Dr. Perlman referred to all over the world to work together on these challenging problems.
And, I was thinking as you were saying that about watching public health messaging through the COVID and the ways that the public, many people in the public have a hard time accepting the fact that science doesn't produce an answer, stop, next question. And this sense of evolving knowledge and discovery, which can sound self-contradictory, but is in fact just continued discovery. So it's just, I can imagine as researchers and doctors in particular, that poses you with a different set of challenges in how you're explaining something, as well as the thing itself.
It definitely does. And I say we have a sort of stiff challenge ahead of us, because people who message on a sort of non-scientific basis are very clear and direct in their messaging, and there aren't nuances the way that there are with science. So as a scientist, if you're trying to give someone an accurate idea, you also have to be sort of conscious, I think, that we may be very comfortable living in a world with some gray areas and some uncertainty, but for a patient or, you know, someone who doesn't think about science all day long, that can come off as not being on solid ground, like Kumar said. And I think it puts us at a little bit of a disadvantage. So we have to think carefully about how we talk to the public and talk to our patients about our scientific findings, what we know and what we don't know.
Yeah, it's interesting. I think that's one of the things that I'll be sort of aging myself with these references to Latour and Popper and Kuhn, but they all wind up being, I think, very significant figures that we all need to learn more about. And one of the things that I'm struck by, I remember when I'm trained as a molecular biologist, and what we used to do as molecular biologists was try to turn things blue. You do some experiment, and all of a sudden, this thing called XGAL would be expressed. Now, and then for a while, we would try to turn things green when it would fluoresce, and then now we would just get a ridiculously large data set and try to model it. So it's definitely changed over time with what we do. But I then became more of a behavioral neuroscientist, where things were much more complicated and with a lot of variables that we didn't exactly understand. And I remember I worked a mentor of mine at Penn was a psychologist named Bob Rescorla, and he was quite a rigorous guy, and he always used the word reliable in describing experimental findings and saying that it had a p of less than 0.05 or less than 0.01 or 0.001 or whatever, and rather than the word that is used more often or all the time, which is significant. And when we do scientific experiments and we analyze them with statistical analyses, they are determining whether if we did it again, we would find the same findings. They're not telling us whether those findings are actually significant. They're really telling us that they are reliable. And it's interesting how when people look at papers and they try to reach conclusions from them, they get made in various ways, as Jocelyn referred to. And it's interesting seeing our students as they develop, because I think they often are challenged with going from the data they have to a conclusion because they're worried about overreaching, but you still have to find a way to reach those conclusions. And I'm further struck when you think about even science itself, that a lot of what we can do most rigorously is demonstrate what's not happening. But everybody wants to know what's happening. And so it's a weird thing where we actually know most about the flip side of a coin, which nobody really wants to see, right? It's just really kind of bizarre.
I sometimes overgeneralize that one of the differences between the sciences and the humanities is the sciences start out with innumerable possible causes and work very hard to narrow down to the reliable result, whereas people in the humanities tend to start with what seems to be an obvious declaration and immediately complicated in every way possible. And that's why we need each other so much, need to be working together. But, you know, what you're all saying does link back in interesting ways to what I think Ted and Kumar and I talked about earlier, which is the change in the way work is being shared among your fellow sciences. And you referred to preprints. And is the preprint the process that y'all were describing to me about putting work up before it's completed for a response?
It's not that it's not completed. We would only put up work that is completed, but before it's been peer reviewed. And if you catch me on a day I just got a manuscript back, I will have strong sailor's language about our reviewers. But on balance, you know, sitting here several days out from my last review, my last judgment was, I think the peer review process is actually overall helpful. And in my own work, they found errors, they found things that helped, things that I wasn't thinking about. And when I review a lot, and now I'm part of that process. So I think that process makes science better. And that's, you know, the public doesn't really get that, right? When something is in the New York Times, and there's a data, it isn't really peer reviewed. It isn't always peer reviewed. I mean, sometimes it is. And sometimes if it's peer reviewed, it can be flat out wrong. It can be compelling to the reviewers, but that process, it's a winding road, and it takes that process. So the peer print is, just to answer your question quickly, before peer review, but when we think it's done.
And there's a series of sites that do that. It was started by the physicists, and there are huge collaborations actually that got this whole preprint process started. And then it expanded in what's called bioRxiv. Now it's in medRxiv and a number of other flavors of this thing. The other part of the preprint process is something that Kumar is better at than I am, which is it's sort of a requirement to tweet the important figure of the thing you've posted on bioRxiv. So that's actually part of the preprint process. And so that's interesting. And there's a group of Twitterati among each of these fields with you want to try to get them to retweet. That makes it a very strange thing, because we wind up connecting to social media and it kind of goes off into the wild blue yonder. And that's part of what's happened with COVID preprints too.
Yeah, this data, the more you tweet, the more cited the paper gets. Just like maybe wrong, but there you go.
Yeah, I see the flip coin or the flip side of that, because in COVID-19 papers, there was so much data that didn't really fit what you were describing, Kumar, where data came out posted well before anyone, even in the original lab, verified that it was true. So I remember early on, in early 2020, there was a paper coming out that said that COVID clearly came from, or the virus clearly came from HIV, because there was an HIV segment in it. And it was a paper. So the good news about it being a preprint is that it was immediately shown to be wrong. It was actually removed in about 24 hours. But the bad news is in those 24 hours, it did a lot of damage. And in fact, there was a meeting called by the emergency NIH meeting, where Tony Fauci wanted to know if this was true and what was going on, which of course no one really could answer except to say this made no sense at all and it wasn't true. So that's the downside of too many, when things are going so quickly, having these preprints followed carefully. A lot of preprints go up that are wrong in fields that maybe aren't so timely, and then there's more time to pull them down and not have them have big effects. So it's not a perfect system having the preprints up.
Well, and I can imagine when you're doing research on COVID and other things that are right in people's view and they're worried about, there's such pressure to publish as fast as you can. It just must be a constant tension.
I don't know if it's to publish as fast as you can. What one wants to do, of course, is if one has important information to get it out as fast as one can. The publication part is less. It's not so easy to publish things that are not considered important, because what did we have? We went from 600 papers a year in coronavirus to 60,000. I mean, it's ridiculous numbers.
So how on earth, if given the number you just cited, and also given, Ted, your comment about the numbers of contributors on one paper, 275 people or whatever, how does this work? And I'm thinking about you, especially Jocelyn, as an assistant professor. How does this work for credit, for shared credit? How's the university coping?
Well, I would say that in general, I think universities have to, and they've been a little slow, but I think they have to adjust their criteria for how we assess someone's body of work, right? So being a middle author on some of these papers maybe doesn't indicate the same thing as being a middle author on, so a paper with 6 or 7 authors, right? If there's only one first author and one last author. Sometimes there's co-authors in those positions on these massive papers. But I think universities have to start to recognize that people are authors on those papers because they make a contribution that allows the science to go forward, right? So I was a co-author on a paper with like 250 authors where we all shared single cell sequencing data from the lung and other tissues so that we could identify where the receptors were for SARS-CoV-2, right? And that paper wouldn't be possible if people weren't willing to take their data from stories that are unpublished or, not quite ready for sharing with the public, and we're willing to put it out there and put it together. So my contribution to that paper is obviously different than the first or the second author, but I think science is just, I don't know, I wasn't here 20 years ago doing science, but it seems much more collaborative and in much larger teams than used to be the case. And so I think the university processes for recognizing that have been maybe a little bit slow to follow.
It's I might say this, as an assistant professor, and Jocelyn isn't saying this, but there's enormous pressure to publish often, and you feel like you're judged by it. And I just recently was promoted a couple of years ago, and I kind of feel that pressure. And really, if I was talking to myself, I would have tried to get myself to calm down, focus on making clear, incremental, solid progress. The publications will sort themselves out. But it's really hard. You feel this enormous pressure because publication is tied not only to university publication or promotion, but also to your ability to get grants and your ability to continue to do that research. Go ahead.
I'll just add, I think that many of us who want to collaborate also have to have our own projects, right? The projects where we are going to be last author or first author or whatever the case may be. And so in my group, I have several folks who are in that assistant professor range trying to make the next step to promotion. And so it's a hard decision to make. Do I take my eye and my focus off my own projects where I know I'm going to be last author and devote some of my time to this collaborative project, which may be more important for public health, may help move this sort of critical area of science like around COVID-19 forward. And so there's always a little bit of a balance there. So I think changing some of the ways that that's measured helps everybody.
Yeah, it sort of used to be, or it still is in some cases, where people are looking exclusively for papers where people are first or last authors. And, that's beginning. I think that is something that really needs to change. I think that there's some metrics we use in a certain sense because they are relatively easy. You can kind of, search that or find it. And part of it is also thinking about what people are doing. I actually find if I'm evaluating somebody for tenure, I start with the research statement that they write and how they put their work in perspective, and also look to more opinion pieces, more review articles that they've also written to kind of give a perspective of their work. A little bit like that broader sense that we talked about, that you were thinking about scientists going from the specific to the kind of broader. And so I think you sort of look at that, and there's other metrics where you can look at, there's some things that people look at in terms of the number of papers you have that have a certain number of citations, but you can also look at the number of citations that you have. And these big papers, so for example, there's also a field we haven't really talked about that's been going for a long time, which is the field of human genetics and the genetics of disorders like schizophrenia or depression or cancer. And there you have had consortiums for quite some while. And when you look at individuals that are in that field, they wind up having a lot of overall citations because these papers wind up garnering a lot of citations. So you can kind of look at a couple of metrics that give you different views of it, but we have to be careful that we don't try to find one and make that the way we do it. And it's, if it is one, it's typically first or last author papers. And that is something that's changed. Science is changing quite a lot. And that is a difficult metric, I think, to continue to be appropriate as a single metric. You can do it, but you also have to think about other ways to look at research productivity.
Well, I would think another interestingly complicated factor would be, a field like the work you're doing, Ted, where you're working on Alzheimer's, where we're so early in the research, as I understand it, or you all are in understanding Alzheimer's, and where, as you talk about always so eloquently, this is about memory, which is about far more than just chemical interactions. This is all cultural, social, relational. So in the case of a scientist working in a field like Alzheimer's that is so still just even trying to define the issues are so complicated. How does that shift the way people think about the work they're doing and evaluation? It seems like it would pose a different sort of challenge than a more established field, or than someone like Stanley who's working on something that there are some parameters and you're working within those parameters trying to solve this immediate problem and getting the resources to do that work at a greater speed than in the past. But that might be my misperception.
No, I think what we do though is find the parameters that we find and use them, very much like a paradigm that Kuhn described in research in astronomy and physics. You have a certain perspective. In Alzheimer's, that paradigm has been A beta and amyloid and APP, and now tau's thrown into it. And that actually has been some bias that has really influenced the research we've done, I think potentially negatively. The challenge with the research that we have to do, so like a disease like Alzheimer's disease, and Kumar needs to jump in here because he's an expert in these as a neurologist. I'll just jump into it. Hopefully I won't hit a mine and blow up. But what I'll say is this: what is most striking about these Alzheimer's is that if someone comes into Kumar's cognitive neurology clinic and winds up leaving with a diagnosis of Alzheimer's, that disease has been developing for 10 to 15 years. And in ongoing experiments where they've developed new tools to see the deposition of A beta and the modification of tau, you can see that in these longitudinal studies. And then the patients have about another decade where Kumar will follow them as they experience a decline. And so that's a 20 to 25 year natural history of a disease, a sort of iceberg that we only see the top of, and all the stuff below the water that was 15 years before somebody came into Kumar's office is unknown. And so the challenge there is to try to get the data that we needed, right? And also data that we don't actually know exactly what we need. And so there's ways to do that. And Kumar's been doing some of this with electronic health records, where you can actually, if you're following people, as that data accumulates. So there's some ways to do it. But I think the thing that's been striking is that we've had to kind of learn how to look back in ways that's made research very difficult. Kumar, I don't know if you want to jump in.
I mean, a lot of things that you said resonated with, what I'm acutely living. What you're describing, this long, humans are long-lived animals, maybe tortoises are longer, but we have disease processes that work over years and decades, and designing an intervention that's going to help over that period of time is actually an enormous challenge. There is no mouse model of, human brains and biology, and coming up with some relevant, small reduced preparation, which you can study, is actually an enormous challenge. And furthermore, even if I was to tell you I have this drug that's really going to work in, one of these diseases that takes years to develop, like Parkinson's or Alzheimer's disease, or even something like diabetes, showing that there's a difference is actually really, really hard. You've got to follow patients for 5-10 years. You've got to look at differences in their overall progression and functional status. And that's actually an enormous challenge. And it's also going to be very, very expensive, because following hundreds of patients for years is going to take a lot of investment. And so, we need new ways of kind of thinking about that problem. Some simple things might be, is there a biomarker like, has gone on in a disease called multiple sclerosis, where you can look at MRI activity, over time? Or is there, something that's going to predict, certain patients are going to be successful or not? And, we have all this healthcare information now, but it's all very, very fragmented. For example, if a patient comes from Mercy Hospital to our hospital, I'm literally looking at faxes, which is like, what? And so, we need a way to, like, each person's health information has to be protected, but it has to be accessible to research. And for instance, during the coronavirus pandemic, there's a TriNetX database. We were able to leverage that to draw insights about which diseases and which conditions were particularly vulnerable to coronavirus. So that was an example of where the healthcare electronic medical systems works for us. But making that work at scale is kind of a challenge that we're going to need to do or figure out as a society. And actually places that have figured this out, like the National Health System in England, are an enormous advantage. and, but they don't represent the world.
Yeah, I would want to add to that. So for things that I think that Jocelyn now and I think about is, so we have these patients who have COVID-19 who are developing long COVID, which may turn into a neurodegenerative disease or some other disease with consequences. And so we're in the position now where in theory we should gather information right now that we can use in the future. But because we really don't know all the manifestations of this disease or how we got there, it's very hard to gather data together to actually know what to measure. Because it would be ideal if we could, because we may have patients who in 5 or 10 years have Parkinson's disease or have something equivalent to the encephalitica lethargica that was seen after the 1918 flu. But gathering those data and really knowing what to do is a challenge. But in a way, it would be exactly what Ted and Kumar are describing, where we could see the tip of the iceberg as it's going into the water, as it were, if we can figure out how to do it.
And the problem is not, well, right now, the problem is not technology. Or ideas. We can think of a million things we could measure, but the problem is implementing it in practicality. How do you take a patient and get them to agree to have the things collected that we want to collect, to know what's going to be, what might be driving any long-term consequences of COVID? So that ability to sort of, I don't know if you could get the answer from big data and say, here's where the most robust signal is in terms of this consequence, and that inform what you collect. Because there's a million possibilities. And so how you put that into practice, I think is the biggest challenge.
And with big data, for that huge paper vomit that Ted referred to, the 17 papers in Nature, I mean, that's big data. And my lab has definitely been, we've been roadkill on the highway of big data. Like our whole research program, 3 NIH grants is a rounding error in one of their plots. And, and when you have big data, your tendency is to blow through things. And really there's gold in those things. There is the answer. If you look at a problem carefully, you have a y-axis and an x-axis, and that's all you need. So there's a challenge in finding data in, or finding answers in big data that are meaningful and that'll make a difference to people, and not sort of blurring everything out in some sort of high dimensional space.
So does that, go ahead, Ted.
I was just going to say, one challenge, and I don't know how much this would really apply, but it's certainly true still with some clinical studies where there's hesitation to do genomic sequencing because of what might be found. I know, and the question is, when you think about some of these disorders, what long COVID might become. Might it become something that looks like Alzheimer's disease, or might it become something that has a psychotic aspect that looks like schizophrenia? Those are all something that people do not necessarily want to know about, right? It's not necessarily, to know that something's going to change, who they are, right? It's a part of the challenge with neurological and psychiatric diseases is we've made some improvement in awareness and acceptance with Alzheimer's and Parkinson's disease, but less so with schizophrenia, and less so when you think about somebody with autism and intellectual disability, in terms of somebody who is going to be accepted and welcomed into all aspects of society. We're beginning to be aware of it, but not necessarily accepting of it. And that means that even if you could get all these people if you had what you wanted to measure, a particular measure, it may be difficult to get everyone to agree to do that because of their hesitancy to find out what might happen. Does that make any sense in terms of possible concern?
Yeah, and it also makes me think of something else. I was talking to another group of researchers, 2 different groups recently, and they were talking about the research they're doing in the areas of water quality and bias built into algorithms was what they call use-inspired research. And so they're listening closely to communities and then trying to design research with those, the community partners in as part of the design process. And in your case, that would be these potential patients. How actively involved how do you all view patients in terms of your research? Are they part of the process of defining problems, or is the science separate from the humans as opposed to statistics about the humans?
Well, in our case, we basically have a Parkinson's Clinical Center of Excellence, and we have several grants that are directly with these patients, and they're really partners in that process. And we actually are planning towards a center grant where we are going to include them as formal members. They will be, at a seat at the table along with the scientists, and they'll have a voice in not only what patient-oriented research we do, but the processes and procedures that go into that. And this is a little hokey, but we try to share our research findings that are done with these patients, with the patients who done. And, having done this, I never planned on doing patient research. I'm a rodent extracellular multi-neuron physiologist. That's how I trained, and that's what I thought I'd be doing for my life. But it's enormously gratifying. The patients want to participate in the process. It's a way that they feel like, listen, I've got this awful disease. How can I give back? How can I be part of the process? And so they really get engaged, and they really geek out on the details. And at considerable, not risk, but it takes effort. They have to drive here. Some of them have to withhold their medications. We even have patients in the operating room, and there's a beautiful article on the ethics of intraoperative neurosurgical research. One of my collaborators, Jeremy Greenlee, is an amazing neurosurgeon, will actually, during our deep brain stimulation implantation surgeries, will record from populations of neurons for a few minutes prior to doing, the clinical procedure. And it's an interesting place to be, but our patients are so excited to be part of this process, something I never planned on doing, because it helps them, I think, I don't know, but I've asked them, they get to be part of this research enterprise. And for them, they become not just some, buddy from, Iowa as a patient with this disease, but they become, part of the solution. And that's an incredibly empowering thing. And something that, we'd like to involve them, as much as possible. We don't send them the data to do analyses, but it's not because they don't want it. So
I think having patients involved is really critical. Obviously, you ultimately hope that your research helps patients, but they oftentimes bring a different sort of perspective, or they help sort of keep your scientific questions in check, right? So, we all know things like cystic fibrosis. We now have therapies for it because we found the gene, we understood the protein, we understood what the protein wasn't doing, and we're able ultimately, why it didn't work, we're ultimately able to develop correctors now, which has really changed the course for patients. And so as a scientist, we're often thinking about what's the mechanism. And for a patient, the question can be totally different, and it can be much simpler. A lot of the patients I see in the post-COVID clinic just want to know that what they're feeling is real. Do other people have this? How common is it? Does it get better? So I think they oftentimes bring a focus that sort of maybe shifts the question that you're asking, or says, wait a minute, I have to take a step back. And instead of worrying about what's the mechanism that drives lung disease after COVID, I have to sort of define it in terms that are maybe more meaningful to this patient. Is this going to get better over time? What percentage of patients 6 months from now are back to doing the things that they want to do? So I think they bring a really important and different perspective sometimes to the questions, how we should prioritize the questions that we're asking.
I will say, as someone who would be more likely to be a patient than in your positions, how grateful I am to hear that this is part of your practice. It's really, that's wonderful. So we only have maybe, sorry, did you want to?
I was just going to comment. I think there's also examples to kind of highlight that. I was involved now some 20 years ago in a foundation called Cure Autism Now, with families and with run completely by families with kids with autism. And in those discussions, we identified the fact that we started the first genetic resource exchange, AGREE, which gave rise to all of the genetic databases that we now have for autism. It was one of the first large-scale genetic studies in neurological and psychiatric disease. And we also realized in our discussions among us that there were a lot of other medical issues that all of our children were facing. that our children with autism weren't just seeing developmental pediatricians, but a cardiologist and pulmonologist and renal specialist and a lot of other doctors. And the realization was that if we could work to develop a better standard of care, we might not be able to cure autism, but we could have better treatment outcomes for individuals with autism, GI problems, for example. That thing called Autism Treatment Network actually bankrupted Cure Autism Now. We had to merge with Autism Speaks because it was so expensive. But Autism Speaks has maintained that and kept doing that over the decades since the merger. And that really started with parents, and it was driven by one of the first hires at Microsoft, who was very well resourced and could finance it, at least in the beginning. And that came from essentially patients, parents of patients in that case, which I think is a great impact, actually.
You know, I would add one other thing. So in some of the work we did before SARS-2, we were working in Saudi Arabia with patients who had Middle East respiratory syndrome. And MERS is a rare disease. And it was very hard to talk to patients. Now, I was working with infectious disease physicians in Saudi Arabia, so I didn't talk to them directly. But one of the problems was that they were so embarrassed about having the disease, and having this conversation helped from these, what I would call, what Ted sort of, he wouldn't use these words, but sort of the tangential issues to the central problem in Ted's case. He was talking about autism. In mine, it would be MERS. We couldn't figure out what was going on because patients just were so embarrassed. They didn't want to really even come to clinic. So just doing that is actually a major intervention that helps all around, both the patients and the investigators.
I don't know if you all know Cynthia Chow, who's in anthropology, a medical anthropologist, but she's also told fascinating stories of work she's done with women in Asia who often are ashamed of having breast cancer and so keep that to themselves until it's too late for treatment. And she's worked with a network of doctors there, really doing sort of feminist conscious raising across a number of countries to try and address the social dynamics of the deadly disease in the ways y'all are describing. So we only have about 10 more minutes, and I'd love to wind up, not to harp on this, but I mean, part of what I see as one of the goals of this series is to give researchers, artists, scholars a chance to imagine what kind of system of reward and acknowledgement, and especially in a world where the tenure system is operating and with its requirements, what you all see. So you've talked about patient, including patients in research, which is another time dimension to the work you're doing. Articles that have 245 authors, which is incomprehensible to me. Working with huge data sets and how that changes the practice of scientific research. Are there questions? And also I would add researchers from different departments that might have very different disciplinary values when it comes to the reward system, and having to negotiate those differences in what's perceived as valuable research. So are there any suggestions or thoughts that you all have about what we could do to strengthen the reward system, the evaluation system, in ways that would help you do the best science possible?
It's a hard question. Maybe, this is something that Ted and me a little bit try to answer in the I&I, but it's really hard to predict who's going to continue to be successful. And I don't know, maybe there's objective metrics on this, but I've been floored when you give smart people who you're impressed with at your faculty and your resources, and you get out of their way and you make life as easy as possible for them to ask fundamental questions. And the rest of the stuff, measurement, right? That stuff is all, I don't know. It seems, I don't know. I worry that it would get in the way of letting these bright people do what they do best, ask fundamental questions.
I want to reinforce what Kumar said. I actually think I think the ideas are really valuable, and we need to not lose track of the fact that they're important. that in the end, what really drives science, what drove science in terms of the COVID-19 vaccines were some gutsy people who thought that you could use RNAs to induce vaccines. And they did that in the 80s. And one of them did that at the detriment of her career. She did not receive tenure at the University of Pennsylvania. And the other person that did receive tenure, he received tenure because he was an MD and a physician. The science was not viewed as high impact when it was happening, and it turns out to have been extremely high impact. But that idea and the fact that those people kept at it, similarly with CRISPR, a technology to manipulate the genome that came out of basic studies in bacteria. I think we really need to realize that actually it's those ideas that make the big leaps, and that's something that we have no idea how to, who's the first author on that or the last author on that. And I think that's something we have to value in the field. We haven't talked about grants. The other thing that we do nonstop, night and day, is write grants and submit grants and bang our head against the wall at NIH. And that's another whole aspect of this, which it's very, very, very challenging in many cases to get that funding, especially if you have good ideas. You have to have the data to really back it up. So I think the thing we have to find is to find the time to develop those ideas and the intellect and the work to value them, the way somebody would think in the humanities of taking the time to read somebody's book to understand what they're saying. Just because what we produce might only be 4 pages in Nature, that doesn't mean that it doesn't have 200 supplemental figures that are associated with that and things we need to try to digest to make sense of. So I think one of the things that I think is important is to find the time, which is impossible to find, to see the ideas in the science that we do, because that's what's going to make a difference decades from now. So what made a difference from the 80s and 90s to now for these mRNA vaccines was the idea that you could do that. And the data came along, they found a company to master it, and then it worked. But it was the idea that could happen that made all the difference.
So I think what you really need is people, and I don't know how to do this well, but you need people who really understand what that person is doing so that you can actually get a feeling for how important it is. When I think of, I remember when the Weissman paper came out, he's worked with Carrico, and it seemed like an interesting idea, but it wasn't really clear what one would do with it. And that was incredibly short-sighted. And they just kept working on it and refining the RNA and making it better and better and making sure it didn't induce responses you didn't want. But recognizing it over the years is a bit of a challenge. Go on.
A large part of their resources actually came from the University Research Foundation at the University of Pennsylvania. In fact, they struggled to get NIH funding. And so the other aspect, I talked about grants, but the other aspect is resources. And I think, in a certain sense, Stanley, what we need are gutsy people to stand up for what we think are good ideas and support the people that can make a difference. That's about trying to see and read the tea leaves and not just count the statistics of citations.
I want to say, Dr. Perlman's I don't know how easy it was, Dr. Perlman, for you to convince people that your work was wildly important in February of 2020, right? But in March of 2020, the stuff that you were passionate about took off. And there might be someone here at the University of Iowa who's studying something else that people are thinking is like some obscure, parasite that they're really passionate about it. And that might be important. And you got to trust it.
In 2003, there were articles in Science, because I've been working on coronaviruses for about 20 years then. And these articles came out talking about how coronaviruses were no longer in the backwaters of biology. And for the 20 of us in the field, of course, I don't know that we knew we were in the backwaters until we were told that we were. But it was certainly a challenge getting our trainees to stay in coronavirology, so that by 2016, I think there was only about 5 or 10 of us in the field, and none of us, except one, was under the age of 50 or 55. So it is a challenge. Now there's no problem.
And Jocelyn, as the person who is probably living the most closely with questions of tenure and promotion, I mean, are there other suggestions you have for what you would like to see in this system of tenure and promotion?
It's a tough question. I think they hit on all of them, right? You need time, you need money, and, those are the things that we all compete for. So I think, probably going back to our conversation about how contributions are valued and making sure that when we're assessing someone's impact, we're really looking at all the ways in which they've had impact and not just sort of, counting those first and last author publications. So I don't know that I have anything new or brilliant to solve the problem, but I think those are the keys.
The other part of that impact is who you mentor and who you teach. So your offspring too.
Well, and your service and your teaching and all the things that we look at, and oftentimes, and it's not just women. There are other people who are asked to serve on committees and maybe do a little bit more service than some, as we're sort of working to make sure that all of our committees are diverse and have various perspectives accounted for. So I think sort of recognizing, like I said, impact in all its forms is important.
Yeah, I appreciate that. And I was about to bring up, if you hadn't, Ted, the issue of mentoring, because I do think that, I think about the difference in having someone like you two senior colleagues as a mentor who thinks in these big very humane ways about your work, and someone who's just driven by the grants or just driven by, publication alone, and how different that would be for a junior person in your labs or a grad student in your labs. So that really thoughtful and time intensive mentoring is huge. And I would guess the other thing that has changed somewhat, back to where we kind of started, is communications. I would guess that many scientists, especially those who lead labs, that you find yourself in a position more and more often where you're having to think about how to explain the work you're doing in terms that someone far outside your specializations, legislators, funders could understand. And that's also a learned process and a very time-consuming one that often goes under the radar, just like service does, as the time spent. This has been absolutely wonderful. I have learned so much from you all. I'm so grateful for the work that you're all doing and how it's serving the rest of us day by day. And I cannot thank you enough. I know that you guys are all incredibly busy people. And so thank you for spending time with me and with the Oberman audience. We'll be recording this to share. And for people who are interested, we'll have another What is Research Now focused on evaluating the arts and new forms of very interesting collaboration in the arts and public-oriented arts, as opposed to more traditional gallery-based arts. So we welcome you. The information's on the Oberman website. But for now, thank you so much. And I hope to see you all again soon.
Thanks so much. Thanks, everybody.
Thank you.
Thanks.